Dear students, as you know that the RNA molecules, they fold onto themselves to make secondary structures. Now we will see what kind of secondary structures are formed as a result of this process. The RNA molecules, they can fold on to themselves due to the complementarity of the bases in the sequence of the RNA molecule. One wonders how many types of the secondary structures can be formed, given that there are only 4 types of the nucleotides in the RNA sequence. It's a very interesting question that we will see in a minute. To represent the primary structure of the RNA molecule, you can simply denote it by 1' as shown in the slide. Also, for the secondary structure, you can represent it by 2' structures. As I just mentioned, yes, it is a very interesting proposition that since the RNA molecules are comprised of only 4 types of nucleotides, there are patterns that exist in the RNA structures. These include the helices, the loops, the junctions, and the bulges. Of course, a bigger RNA molecule comprises of these 4 types of structures brought together to make the bigger RNA molecule. To begin with, as you know that if an RNA molecule were to be unfolded, it would start with a 5' end until the 3' end, as shown here. Each of the nucleotide base can be represented by a stick, as highlighted here. Once the RNA molecule starts to fold, the 3' end here, it shifts towards the 5' end here and makes a molecule. So essentially, a 3' end will come together with the 5' end. This will form a secondary structure that will continue on to be folded further and hence provide a functional property to the RNA. Okay, so moving on, the second type of the RNA secondary structure is the helix. As I just mentioned, that the helix can be formed once the 3' end of the molecule comes towards the 5' end. Of course, on this side, the RNA molecule can continue to form other secondary structures. Dear students, it is important to note that there is a difference between the RNA helix and the DNA helix. In the RNA helix, the molecule folds on to itself. So if the molecule were to be represented by a rope, the 2 ends of the rope, they come together and then bondings are formed between the inner portion of the rope. The second 2' structure for the RNA that we will consider today is the hairpin loop. It is very interesting because a hairpin is exactly what it looks like. In this type of structure, the 3' end comes together with the 5' end, as we just saw. However, some nucleotide bases, they cannot come together as they are not complementary. An example of that would be a G with an A, a U with a C. So of course, U is not complementary to C and G is not complementary to A, and this will result in the lack of bonding between these nucleotides. So if these nucleotides are not bonded together, they take the form of this big hairpin I. The rest of the structure continues to fold accordingly. To make it more clear, let me write a sequence for you. This will constitute a hairpin loop. The RNA secondary structures, as I just mentioned, can take more forms as well. One thing to remember is that once the hairpin loop forms, then the eye of the hairpin must at least comprise of 4 nucleotides in order to avoid the steric hindrance between the molecule strands. And that the hairpin loop reverses the chemical direction of the RNA molecule.